Hello viewers, you're welcome to this lecture series on protozoans. I have earlier in a video class defined protozoans to be the unicellular organisms that are eukaryotic and exhibit heterotrophic nutrition. They are animal-like organisms. And we've also looked at the various types of protozoans, and that we have and they are classified into four groups, or they are grouped into four categories based on locomotor apparatus, and there are 10 phyla in this subkingdom Protozoa. Based on locomotor apparatus, they have been grouped into the amoeboid protozoans that use cytopodia for locomotion, the flagellate protozoans that use flagella, and the ciliate protozoans that uses cilia for locomotion, and the ones that do not have locomotor organelles, the sporozoans, which are also spore forming. So I want to look at the flagellate protozoans because in our earlier video we had looked at the amoeboid protozoans. Now, what are flagellates? The flagellates are organisms which have one or more whip-like organelles called flagella. They may be solitary, colonial, free-living, or parasitic. They may be solitary, colonial, free-living, or parasitic. The solitary ones live alone. The colonial ones form clusters, while the free-living ones are seen swimming in the water column or wherever they find themselves. Some are parasitic. Then parasitic forms live in the intestine and bloodstream of the host. They live in the intestine and bloodstream of their host. An example of parasitic flagellate is Trypanosoma brucei, which has an interesting life cycle as it uses to host humans and fly. Trypanosoma brucei is a protozoan, a flagellate protozoan that causes African sleeping sickness known as trypanosomiasis. And the vector is fly, and is transmitted to humans by the bite of the fly, which eventually causes the disease condition known as trypanosomiasis. Many other flagellates, like dinoflagellates, live as plankton in the oceans and freshwater. Some flagellates are autotrophic, while others are heterotrophic. Some of the flagellates are autotrophic, while some are heterotrophic. The flagellate protozoans belong to four phyla. They are the phyla include the phylum Dinoflagellata, the phylum Euglenozoa, the phylum Parabassalia, which are also called the Parabassalis, and the phylum Diplomonada. Four phyla of protozoans are grouped into the flagellate protozoans because of their possession of flagella. They possess flagella for locomotion. These organisms possess flagella for locomotion. So we want to look at the first phylum, which is the phylum Dinoflagellata. We want to look at the dinoflagellates. Now, dinoflagellates, okay, we want to look at the first phylum, Dinoflagellata. The first phylum, Dinoflagellata. Now, before we look at the first phylum, Dinoflagellata, I want to look at one very important feature of these organisms. Now, the phyla Euglenozoa, Parabassalia, and Diplomonada belong to a supergroup of protists called excavata. They are called the excavate. Excavata are defined by an asymmetrical appearance with a feeding groove that is excavated from one side. Now, the phylum Dinoflagellata. The phylum Dinoflagellata. What are dinoflagellates? Dinoflagellates are a large group of flagellate eukaryotes that are known to be photosynthetic, but a large fraction of these are in fact mixotrophic, combining photosynthesis with ingestion of prey or phagotrophy. So most dinoflagellates are known to be autotrophic. They are known to be autotrophic. They are able to carry out they are able to manufacture their own food. But some combine photosynthesis with phagotrophy, and that is that kind of combination of feeding habits, of such feeding habits of combining photosynthesis with phagotrophy is called mixotrophy. So they exhibit mixotrophy. Some species are endosymbiotes of marine animals and play an important part in the biology of coral reefs. Some are found within marine animals, and they exhibit endosymbiosis with such organisms. Other dinoflagellates are unpigmented predators on other protozoa, and few forms are parasitic. For example, Odonium and Cysteria species are parasitic species, while some are predators of other protozoans. Some dinoflagellates produce resting stages called dinoflagellate cysts or dinocysts as part of their life cycles. They produce dinocysts. Dinoflagellates are unicellular and possess two dissimilar flagella arising from the ventral cell side. And such flagellation is called dinocont flagellation. They possess two dissimilar flagella. They have a ribbon-like transverse flagellum with multiple waves that leads to the cell's left, and a more conventional one, the longitudinal flagellum that beats posteriorly. So they have two flagella, one is transverse, one is longitudinal, and that's what this figure is illustrating. Now, the transverse flagellum is found within a groove. It's found within a groove, and it beats to the cell's left. And we have a longitudinal one that extends posteriorly, which propels the cell forward. And they have a cellulose armor. Some of them have cellulose armor. A bloom of certain dinoflagellates can result in a visible coloration of the water, colloquially known as red tide, which can cause shellfish poisoning if humans eat contaminated shellfish. Some dinoflagellates also exhibit bioluminescence, primarily emitting blue-green light. Okay, they exhibit bioluminescence. Bioluminescence basically refers to a phenomenon in which living organisms produce light. When living organisms emit light, it's called bioluminescence. And the dinoflagellates emit a blue-green light, usually at night. They emit a blue-green light, and that's what this figure is illustrating, where a blue-green light is observed in the waves, that are produced in the ocean. Now I want to look at the phylum Euglenozoa, or the Euglena. The phylum Euglenozoa. Euglenoids are protozoans that are colorless or green with two flagella arising from the anterior recess. Now the dinoflagellates have two flagella, they have one longitudinal flagellum and one transverse flagellum. Okay, but in the case of the euglenoids, the flagella are placed in an opening, an anterior opening or recess, and they also possess two flagella, just like the dinoflagellates. About 1/3 of the approximately 40 genera of euglenoids have chloroplasts and are fully autotrophic. The others lack chloroplasts and they ingest their food and are heterotrophic. So some dinoflagellates possess chloroplasts and are able to carry out photosynthesis, while some are heterotroph, they engulf their food. A number of euglenozoa have acquired chloroplasts through endosymbiosis. That's a symbiosis in which one organism lives inside the body of another, and both function as single organisms. Typical examples of protozoans in the phylum Euglenophyta or Euglenozoa in this case include Euglena viridis, Euglena gracilis, and Chlamydomonas species. Now here are pictures of these organisms. Figures showing, this one is showing Euglena viridis, and this Euglena viridis have both plant and animal characteristics, and that's why among the protists they are classified both as Euglenophyta, and among the Protophyta, the subkingdom Protophyta, and also in the phylum euglenozoa among the protozoans. All right, the animal-like features it has the stigma, which is also called the eye spot for slight vision. It detects light, and then they have two flagella for locomotion, and they do not possess cell walls. So they are, in this regard, known to be animal-like. And then they also have plant-like features in that they have chloroplasts for photosynthesis and large contractile vacuoles. The same thing is applicable to Chlamydomonas, which has chloroplasts and also possess eye spot, stigma, and the flagella for locomotion. They possess flagella for locomotion. We want to look at the phylum Parabassalia. The phylum Parabassalia. The Parabassalis are anaerobic flagellated protists which contain at least one parabasal apparatus consisting of a parabasal body, that's the Golgi apparatus, and a parabasal filament. They possess parabasal bodies and they are anaerobic. Parabasal bodies such as Golgi apparatus. Parabassalates or parabasal means found near the nucleus, and kinetosome. The kinetosome is a structure in the flagellate protozoans which forms the base of the flagellum, consisting of a circular arrangement of microtubules. Their basal bodies are linked to parabasal fibers that attach to the prominent Golgi complexes distinctive to the group. They are within the supergroup Excavata. Parabassalate can be distinguished by the presence of the microtubular axostyle and many flagella. Now, the distinctive feature is that they possess an axostyle and also possess many flagella. Unlike the flagellates and the euglenas that have two flagella, the parabassalates have many flagella, more than two flagella. And axostyle is a sheet of microtubules found in certain protists. The flagella are arranged in one or more clusters near the anterior of the cell. Parabassalates move with flagellar and membrane rippling. Trichomonas vaginalis, a parabassallate, can cause sexually transmitted diseases in humans, employing these mechanisms to transmit through the male and female urogenital tract. So, Trichomonas vaginalis is able to move through with the aid of flagella and membrane rippling. It moves with the aid of flagella and membrane rippling. T. vaginalis or Trichomonas vaginalis causes trichomoniasis, which is a sexually transmitted infection, especially of the vagina, marked by persistent discharge and intense heating. They're able to cause trichomoniasis. It also causes erythritis in males. It also causes erythritis in men. Now, here is a figure showing Trichomonas vaginalis, which has 4 flagella at the anterior end and 1 at the posterior end. That's 5 different flagella. And they also have a cytostome, a mouth that's like a groove. And they also have 1 flagellum attached by the undulating membrane. Okay, another flagellum. So you have 1, 2, 3, 4, 5, 6 flagella. You have 6 flagella. Parabassalates are mostly symbiotic. They are mostly symbiotic, living in hosts ranging from humans to termites and cockroaches, enabling them to break down the cellulose and the dead plant material they eat. These symbiotic parabassalates, in turn, have symbiotic bacteria that aid in locomotion and cellulose breakdown. Parabassalates are anaerobic and lack mitochondria and peroxisomes. They are anaerobic and they lack mitochondria and peroxisomes, but have specialized organelles called hydrogenosomes, in which anaerobic metabolism takes place. They possess hydrogenosomes instead of mitochondria and peroxisomes. They lack feeding grooves found in most other excavates. The parabisalids lack feeding grooves that are found in other excavates. These organisms carry out a special type of closed mitosis called cryptopleuromitosis. They carry out cryptopleuromitosis, characterized by the persistence of the nuclear envelope and the presence of an extra nuclear spindle. So cryptopleuromitosis is common among the parabisalids. It's a kind of mitosis in which the nuclear envelope does not disintegrate in prophase or pro-metaphase. The nuclear envelope remains intact, but it splits into the two parts that we found in the two daughter cells. The next type of flagellate protozoans we want to look at are the Diplomonada, those in the phylum Diplomonada, the Diplomonads. Among the excavator are the Diplomonads, which include the intestinal parasite Giardia lamblia. Okay, the Giardia lamblia of this phylum Diplomonada. They lack both mitochondria and Golgi apparatuses. The Diplomonada lack mitochondria and Golgi apparatuses. However, they are known to possess modified mitochondria in the case of Giardia lamblia called mitosomes, but these mitosomes are essentially non-functional. They are not used in ATP synthesis the way mitochondria are, but are involved in the maturation of iron sulfur proteins. So they are actually non-functional forms of organelles. Diplomonads exist in anaerobic environments and use alternative pathways such as glycolysis to generate energy. Okay, they are found in anaerobic environment and uses glycolysis instead to generate energy. Each diplomonad cell has two identical nuclei and uses several flagella arranged symmetrically about the body's main axis for locomotion. They have several flagella. So the diplomonad and the parabisalids have more than two flagella, while the euglenoids and the dinoflagellates have just two flagella. Some particularly unpleasant flagellates are contained in the genus Giardia, which are intestinal parasites. One species, Giardia lamblia or Giardia intestinalis, causes intense nausea, cramps, diarrhea, gastrointestinal disorders, and malabsorption in its human host, a condition that may take several months to dissipate. It inhabits the duodenum and the small intestine. The infection is readily transmitted through drinking water contaminated with infected feces. Now, this is Giardia lamblia. As said, it has more than two flagella for locomotion. We have 1, 2, 3, 4 flagella in this case in Giardia lamblia. Thanks a lot for watching this video on the flagellate protozoans, and we urge you to watch more videos, especially as I present in the next video the ciliate protozoans and subsequently the sporozoans. Please subscribe to my YouTube channel to receive updates of various videos that will be released soon.